So thank you for this introduction. It's an honor to be with you today. I will talk about DPP-3 in cardiogenic shock. So as Noma said just before, DPP-3 is a zinc-dependent metallopeptidase, mainly cytosolic, but also detectable in the circulation. And the abbreviation for it is CDPP-3. CDPP-3 cleaves and terminally active biopeptides such as enkephalin, endomorphine, and angiotensin. It modulates the angiotensin renin system. If we look in patient cardiogenic shock after myocardial infarction, patients who had higher levels of CDPP-3 at admission had higher mortality rates. So DPP-3 appears to be a prognostic biomarker in cardiogenic shock. To understand more, we want to evaluate the cardiac expression and plasmatic levels of DPP-3 in a cardiogenic shock model in a large animal. In collaboration with Professor Ouattarassim in Bordeaux, we've developed a model of cardiogenic shock in sheep involving the creation of an infarct by intracoronary injection of alcohol in first intention in the IVA coronary. The sheep are anesthetized, then catheters are placed with a Swan-Ganz catheter and Miller catheter to have PV loop. Then the induction of shock by injection of intracoronary of alcohol, followed by 1 hour of stabilization before 3 hours of follow-up. Then the sacrifice, the heart explantation for tissue analysis. Throughout the procedure, blood samples and echocardiographic are taken repeatedly. Our shock is defined by a 30% drop in cardiac output or mean arterial pressure associated with a lactate levels more than 2.5 millimole per liter. They receive noradrenaline perfusion if the sheep need it. So we want to study DPP-3 expression in the heart and activity of DPP-3 in the plasma in this model in 4 groups: a sham group, a shock group where animals had only myocardial infarction, a CLS group where animals had myocardial infarction plus ECLS, arteriovenous ECMO, and the last group was ECLS-DOBU for animals who had myocardial infarction, the ECMO, and infusion of dobutamine. The time points are represented in the figure. We have baseline 1 for just after general anesthesia, baseline 2 the induction of shock, and the 3 or 4 hours follow-up. 3 hours for the sham and the shock group, and 4 hours for the ECLS and DOBU group, according to the levels of assistance we adjust from the base flow rates from 25% to 100%, and we increase in each step 25% of the flow. So here we have the mean arterial pressure and the lactates, and we can see that mean arterial pressure and lactates level are stable for the sham group. So it's indeed a control group, a real control group. And for the 3 other group, we have a decrease in the mean arterial pressure and an elevation of the lactate level. So it's a cardiogenic shock group. If we looked at the activity of CDPP-3 in the plasma, we see an increase of the activity in the ECLS-DOBU group and ECLS at H4. Which is surprising is that the levels of the sham as the same as the shock. We looked at DPP-3 expression in the heart by using Western blot in the left and the right ventricles and in the septums. And total heart corresponds at the 3 previous sections. And we see an increase of DPP-3 expression in the right ventricle and in the septums with significant only in the ECLS group. We looked at the association between CDPP-3 and DPP-3 in the heart, and we found no correlation between the activity of DPP-3 in the plasma and the expression in the heart. But we cannot conclude about a potential release from myocardium to the plasma with our results. So our result demonstrated an increase of DPP-3 expression in the heart. So which can be explained by de novo expression because the duration of the model is too short. So this data suggests that cardiac DPP-3 does not seem to come from the heart and might come from other cells and maybe inflammatory cells. We know that there is an early inflammation following a heart attack. So what is the contribution of inflammatory cells and DPP-3? We studied CD68, a marker of inflammatory cells, the expression in the heart by using Western blot in the same part as we did with DPP-3. And we found a significant and positive correlation between the expression of CD68 and DPP-3 in the heart. So the increase of DPP-3 in the heart seems to originate from immune cells or CD68 positive cells. So the limitation of our study is the model himself, because there is an infarct, but there is no reperfusion. The short time, maybe maybe the elevation of DPP-3 for the shock is after the 3 hours. And for the surprising results about the sham, we will add more animals. For the perspective, if DPP-3 is brought to the heart by inflammatory cells, what are they? It requires further study to know that. We will evaluate counts of levels in plasma and the transcardiac gradient of the activity of CDPP-3. And what are the consequences of the elevated DPP-3 in plasma in this model? We want to evaluate cardiac function and plasma of CDPP-3. And if we found something, it will be interesting to modulate the activity of CDPP-3 in this model. Thank you for your attention.
Questions? If not, I'm not an expert on experimental models, but why did you split the animals in a group with ECMO, another with dobutamine? Why not? Which is the rationale for this?
Because maybe it is a different group between ECMO, maybe more inflammatory with the ACLS than without the ACLS, and the dobutamine may be more inflammatory or make the difference on the DPP-3 levels. We don't know about that, and we want to know if there is something different between the group and between our patient, because we have the severity of the patient is different than one without and one with, but all the sheep are the same severity, but we want to know if the ECMO in itself make the difference.
All were in cardiogenic shock.
In cardiogenic shock. And the same for the dobutamine, for the catecholamine.
Okay, thank you very much, Claire.